Okay, so cellular specific immune responses here, also known as third lines of defense. Okay, so first line, second line. So second line always initiates the third line of immune response, okay, or defense. So in this case, we call adaptive. Adaptive means that your body have to learn. It's not innate. Adaptive basically something like the skill that you have to learn. That's not I say that innate. Innate means something that you do not need to learn. You automatically know what to do. But adaptive means something that you need to learn. For example, writing, okay, you need to learn, okay? So in contrast to non-specific immune mechanism, they're always present and ready to respond to infection. Specific immune response develop in response to an infection only and adapt to a specific pathogen. So they learn from this. Okay, so they take time. Innate, you don't need to take time. Already prepared, already there. You already know what to do. That's innate. Okay, you don't need to train. But this one you have to train. So that's why it takes longer period of times, right, to complete the entire process. Okay, while non-specific immune response destroy pathogen and prevent the spread of infection, the body mobilize its specific immune response. So it takes a few days to activate a specific immune response, but once it gear up, then the mechanisms are extremely effective. You can see that you have a lot of clonal expansions and a lot of this lymphocyte help us to fight with the pathogen. And very, very important, they only respond to a specific antigen. And this is the powerful of the immune response. They do have this, what we call the immunological memory, right? Immunological memory. So they can remember who is the pathogen. And it come in the second time, I don't need to check, okay, are you a pathogen? Are you a blah, blah? No need. Straight away, they're going to remove the pathogen before the pathogen causes the problems or any symptom to our body. Okay? So we have two types of specific immune response. We are the cell-mediated immunity, okay, or an antibody-mediated immunity, okay. So cell-mediated involve T lymphocyte, and antibody-mediated involve B lymphocyte. Okay, so therefore we can see that two key cell types that participate in specific immune response, they are lymphocytes and also the antigen presenting cells, which are macrophage. Just here. Okay, clear? Any questions? Okay, so now what we're going to do here, we're going to continue with the lymphocytes. Now, before we continue lymphocytes, I will start recording a while. So now I have summarized everything just now. So here the photo, everything. So you guys have taken a photo, either copy downs or you have the I see that screenshot it. So lymphocytes, they're important in both specific immunity. We have two types, T lymphocyte and B lymphocyte. We develop in the bone marrow because in the bone marrow we have these hematopoietic stem cells. Okay, so only mature lymphocyte they can carry out immune response. So means that they have to complete their development. So either they complete the development in the bone marrow, then it become B cells. If they complete in the thymus, then it become T cells. So each type of mature lymphocyte is specialized to respond to one antigen only, as I said just now, right? Okay. So once they are matured, we have done the selection, everything. So they will reside in the lymph organs, including the spleen, lymph nodes, tonsil, and other lymph tissues, strategically positioned throughout the body. The lymph nodes are particularly, so it means that they are there to check, something like how to say that, checking, okay, the presence of the pathogens or not, okay. So you can see that in this diagram, so bone marrow, because of the hematopoietic stem cells, so we have the stem cells here, then develop into the lymphocyte. So you can see that this lymphocyte, they cannot carry out any activity yet. If they migrate to the thymus, so it will form T cells. If it remain in the bone marrow, it form the matured B cells. So in the thymus glands, thymus make T cell become immunocompetent, so means that they can have the immunological response. Why? Because developments of the T cells receptor here. Okay, so what's the T cell receptor? T cell receptor, they are specific surface protein with distinctive receptor site. So this receptor site also known as antigen binding site. Okay, it's also known as the antigen binding site. So the T cells populations will undergo positive and negative selections. So what's mean negative selection? So T cell in the timers that react or against or target the self-antigen, we will send them for apoptosis. Apoptosis basically means that program self-death. Okay, so it means that we want them to die. We program them to die. Why? Because if we release them from the timers, then they're going to attack our own cell. Because the formations of the T cell receptor is a random process, you may accidentally, okay, form, okay, the T cell receptor that can target our self-antigen. Okay, so for example, I ask you, okay, you simply shout out the number, randomly give me number 1 to 100. 1 to 100, give me one number. Okay, so out of 1 to 100, some of the number belongs to someone else. So if you shout a number, you may accidentally hit it because it's a random process, okay, by chance. So no problem, we can have this negative selection. But if the T cells that can recognize the self-antigen, oh, this self-antigen we don't bind, and bind only with the non-self-antigen, so these T cells that are allowed to continue to mature. Are you clear? So this, they're measured to what? They measured into the cytotoxic T cells or helper T cells. So how we know? We check their co-receptor. So cytotoxic T cell will be CD8. Okay, helper T cells will be CD4 co-receptor positive. Okay, so after positive selection, T cell differentiates and leave the timers to pick up the residence in other lymph tissues. Okay, so it means that now by doing this, we confirm that our T cell distinguish between the self-antigen. Oh, cannot bind. And non-self-antigen, yes, can bind. Okay, so two main populations, they are CD8 cytotoxic T cell or CD4 helper T cells. So what is the function of cytotoxic T? Cytotoxic T cells, they are formerly known as killer T cells, but in our syllabus, if we use the word killer T cells, no wrong, yeah? Okay? So they are also known as CD8 cells because they have a glycoprotein and designated CD8 on their plasma surface. Okay, now what does this mean? So if a cytotoxic T cells, just now I have drawn. Okay, this is a cell. So with the nucleus, the large nucleus, so they have the T cells receptor. So this T cells receptor, okay, let me label it. T cell receptor. So beside this T cell receptor, they do have the additional proteins, something like a log like this. So this log is known as the co-receptor. Okay, co-receptor CD8 because it's the cytotoxic T cells. We will know about this later when we talk about antigen presenting cells. Okay, they have this co-receptor CD8. So they have TCR. So in this case, we know that there are definitely T cells. Or what kind of T cell? Because of CD8, so they are cytotoxic T cells. So what they do, they recognize and destroy the cells with foreign antigen on their surface. The target cells include, okay, infected cells, cancer cells, or foreign tissue graft. Now, what's a foreign tissue graft? Foreign tissue graft basically means that the tissue transplant, for example, kidney transplant, lung, organ transplant, those because they're from other person. So it means that they can recognize those non-self antigen and destroy the graft, which is bad. Okay, because we have organ transplant, we want with the idea that we want to save the life, but now our body immune system actually reject it. Okay, organ rejection. Sorry, the graft rejection. Then we say that. Okay, so how they destroy? How they destroy the cells? So they secrete toxic chemical. I will tell you guys this toxic chemical. I mean, the next lessons, not now. Okay. Now, how about helper T cells? Helper T cells, also known as CD4 cells. So means that if you look at this, this I label it as a cytotoxic T cell. Now, CD4 cells, what's so different? Same thing. If you look at it, they are almost the same. They also have the T cell receptor. They also have the co-receptor. Can you see that? So we will label this also T cell receptor. They have the co-receptor, but this is CD4 co-receptor. So this one become helper T cell. They're specialized. Okay, so what they do? So they're also known as CD4 cells. They are CD4 cells. They have surface glycoprotein designated CD4. So they help to secrete cytokine. Okay, highlight the word cytokine. So this cytokine actually going to activate B cells, T cells, and also macrophages. Okay, so after an infection, you will have a memory T cells, both memory cytotoxic T and memory T helper cells remain in the body. So this is for the T lymphocyte. How about B cells? B cells produce in the bone marrow daily, and also they are genetically programmed to encode the B cell receptor. So this B cell receptor is an antibody. Again, formation of B cell receptor is a randomization process. Okay, you wouldn't know what kind of the antigens they are going to target. They may target self-antigen, they may target non-self-antigen. So most important thing is we need to choose the correct, okay, what you call this, clones. Okay, so when the B cell come into contact with the antigen that bind the receptor, B cells now become activation, and this activation have to be completed by the participation of helper T cells, because helper T cell actually release the cytokine. Clear? We release the cytokine. So these cytokines will be received by B cell in order to be activated. Once activated, B cell will rapidly to form clones of identical cells. So in this case, we form the clone. So the name here called clonal expansions. Okay, so clonal expansion. So in this case, you have one cell, one B cell. So develop into many B cells. 1 to become 2, 2 become 4, 4 become 8, and so on. So some of them become plasma cells. Some of them become memory B cells. Are you clear? So what's the function of plasma cells? Sorry. So plasma cells, the cells that produce and secrete antibody. Okay, so this antibody actually bind to the antigen that originally activated the B cells. Memory B cells, they are long-living memory B cells that remain in the circulating system. Okay, they proliferate rapidly and develop into the plasma cells again, and more memory B cells during the second invasion. So this one basically means that we repeat the cycle, but with a short period of time, because we are trained already. So try to imagine, before that you do not know how to cycle. So you need to train to cycle. You take few months to learn. But once you know the skill already, even though few months you didn't cycle, but immediately ask you to cycle back, you can cycle. Okay, or you can swim in this case. So why? Because you already know, you already learn in this case. Okay, so means that you're going to do the thing fast. Okay, so therefore pathogen can be destroyed and removed before any symptom. So no symptom at all. Okay, you wouldn't know actually you are infected. Are you clear? So similar to the vaccinations idea, vaccinations, for example, you get a COVID vaccinations. So after vaccination, you might be infected, but you wouldn't know. Why? Because your body actually straight away produce the antibody and do whatever means background scenes, behind the scenes, to help us to remove the pathogen. Are you clear? Okay. So that's why the symptom will be very mild or even no symptom. Okay. Doesn't mean that you won't be infected, guys. We still will be infected, but you may not have any symptom. Okay. So again, this is a summary. You can see that hematopoietic stem cells from the lymphoid progenitor or lymphoid stem cells. So it depends. It differentiates into the B cells or T cells. So it develops. So in the bone marrow, you can see that B cells start to form the B cell receptor. So if wrong, they will go for apoptosis. If wrong, go for apoptosis. So means that we make sure that the B cell that produce the matured B cells only targets the non-self antigen. Same goes to the T cells. You can see that we have the T cells who start to form the receptor. Can you see that? T cell receptor. And also what we call the co-receptor here. So if the T cell receptor bind to the self-antigen, we send them for apoptosis. Okay, if cannot, we send them apoptosis. And they have these two co-receptor. So right now, for CD8 helper T cells will be CD4 co-receptor. Killer T cells or cytotoxic T cells will be CD8 co-receptor. We're going to look at the CD4 and CD8. Why it's so important to know about CD4 and CD8, that make sure that we differentiate between the helper T cells and killer T cells. So you can see that we have the positive selections until we get, or negative selections when we send the cells to die. Are you clear? So this maturation process for T cells inside the thymus, B cell actually in the B cells, bone marrow. Okay, so with this, I have done for the lymphocyte.